getting older now. Just like just yesterday you graduated, but
You got married.
yeah, I know, right? Syria.
Really have to go to work or something.
Why something on my mind these days, you know? Like for the past couple of weeks, my father acted real strange. And like, wait, forgetting like bring the keys and all that kind of thing, but it's like, you know, just getting to service.
So what are you doing now?
It's like, just last week, he drive past the house, you know, like if you're not even living there. And just yesterday, or day before, he forgot my sister's name. It's like, he looking at her and he didn't even recognize her. And she just stand up there and it's like, she had shocked as to how her father could recognize her. And she just had to cry with it. Well, that's exactly what I want to talk to you about, because we came in by the doctors today. And we still in the hospital and they run some tests on him. And they say that they think it might be Alzheimer's disease. But that's why I want to ask you about our colleague, because I know your father is a doctor. Yeah, because you know, me and this here, you know, we just want to get understanding of it, because we can't just, it's like this thing just, so we want to get some information on it, and you know, as your father doctor, I wonder if you could.
We'll see.
Yeah, wait.
Well then, right?
Yeah, wait.
Oh, okay.
That is Mark, right?
Of course I know Mark. And I heard he got married to this lovely young lady.
Hi, good day.
Hi, you must be Mrs. Shakir.
Yeah.
Okay, nice to meet you.
Same place.
So, John told me why you are here. And first of all, let me say that Alzheimer's disease is something really difficult to go through. And especially when you have a loved one involved. And it's sometimes hereditary. And there is no known cure for it.
Lord!
Oh, you gotta tell the children.
And the grandchildren will get,
What grandchildren?
but they're going to be married recently.
Go to one thing at a time.
Let me stop you there. I think you might be a little bit misinformed. But I think I have just the thing. Well, there's this YouTube channel called Biochem JM.
Really?
Really. And there is a video tutorial there showing a lot about Alzheimer's disease, and I think it'll be good for you all to look at.
Okay.
So, you all ready? Director, run the
Gosh. Right.
Everybody, and this is the video tutorial for the video code entitled Recall Relapse. Content of the tutorial is subdivided into 5 sections, which are what is Alzheimer's disease, how does it affect brain, protein backgrounds, amyloid precursor proteins, and neurofibrillary tangles, and tau. Hope you like the video.
Hi everyone, and today we're doing a video tutorial on Alzheimer's disease. And so, you know, most of us might know someone, or some of us might know someone who suffers from this disease. And so, knowing about the biochemical basis behind it might be beneficial to some of us. So let's get into Alzheimer's disease. Firstly, we want to know what exactly it is. Well, basically, it is a neurodegenerative disease of brain cells. What it means is that there is a loss of brain cells due to death and degeneration. Now, it is the most common form of dementia found in humans, which means what dementia is an impairing or loss of brain function. Now, here we have the stages of Alzheimer's disease listed from 0 years to 8 to 10 years. So from on top here is the initial symptoms, and we're going down to see how the symptoms exacerbate as the years go on. So firstly, you might find the individual having or displaying some form of short-term memory loss. Now, memory, short-term memory is stored in the hippocampus. So this is usually the first region of the brain that is affected by Alzheimer's disease. Next, there's a loss of logical thought or logical thinking. Sometimes the person may not be able to do simple maths and so forth. And this is found in the frontal lobe. So here the disease progresses towards the frontal lobe of the brain. Next, there's a loss in emotional control or regulation. Sometimes the person may be given to bursts of emotions. Sometimes they will not be able to control anger or sadness or even happiness. Now, there's a loss in perception of senses. So the person may not be able to see well anymore. They may not be able to hear or taste as well as they used to. Now, going down to around the 8 to 10 year range now, where near to the end of the person's probably life, you find that the oldest memories are beginning to lose. So they don't remember how they were when they were children, and they will not remember certain important events in their life. And this might be a really hard time for family members. And usually the final symptom is loss of involuntary muscular function, which basically means that the person may not be able to live anymore, as the heart may not be able to beat anymore, or there will not be, the lungs of the person will not function anymore either. And so this is what usually kills the person who is suffering with Alzheimer's disease.
Okay, so now we'll be looking at how does Alzheimer's disease affect the brain on a macro scale. And a healthy brain actually functions when there is no inhibition of electrochemical signals from one neuron to another. And here actually briefly explains how that occurs. One neuron would have an electrochemical signal, will be converted to a chemical signal and then into an electrical signal again. And that's how you actually create memories, which is actually loops or circuits in the brain from one neuron to another. And that's how you create memories, via a specific pathway. And in between two neurons, there is something called a synapse, which is a space, and neurotransmitter chemicals actually flow from one to the other. So here we actually have a simple diagram of three neurons in a brain, and we can see the the messages going from one to another. And what's characteristic of a brain that's affected by Alzheimer's disease is actually the presence of insoluble proteins. And there are usually two, which is the beta amyloid plaque and the neurofibrillary tangle. And what's the actual problem is that they're insoluble. And the brain is, of course, a soluble environment. So, on to the other part now. Which is a general background in case you're not aware of simple protein interaction. So a protein is a polypeptide, meaning it's made up of amino acids which are linked together by a peptide bond. And it's important that they have a primary, secondary, tertiary, and quaternary structure. Now what does that mean? Biochemistry is three-dimensional, meaning that what you see in the textbook is a two-dimensional frame, but what you need to realize is that the three-dimensional structure of proteins is very important. Now, this actually is a simple diagram of some amino acid residues linked together by a polypeptide bond, and what usually happens is that they must be folded into secondary and tertiary structure and whatnot. And chaperone proteins usually have that. Chaperone literally means that they supervise or they help. And usually, most of the time, a protein is folded correctly, and then it goes on to provide its relative function. So it might be a transmembrane protein, or it might be keratin, or whatever function that it's supposed to provide. But actually 30% of the time, a protein is actually folded incorrectly. So instead of a sort of, you might have a microfibril or whatnot, it might get changed into something that it's not. And the body would actually degrade it and then take its components and reshape it using a chaperone protein again so that it could perform its correct function, because the body doesn't want to waste those amino acids that went into it. Now, most of the times, the chaperone protein is able to correctly amend this problem, but sometimes it mightn't be able to do anything, and that's actually where you have conformational diseases, meaning protein conformational diseases, because of course the three-dimensional folding is the protein conformation. That's what we're talking about, and this actually happens in Alzheimer's disease, where Alzheimer's disease is described as a conformational disease, and it's because of incorrect protein formation. Now, proteins must exist in a soluble environment because most of our body is 70% water, and proteins, a lot of them are insoluble, and the phospholipid bilayer is what we'll be talking about next. The first example of Alzheimer's disease and protein misfolding is with regards to amyloids and APP. Now, an amyloid is an insoluble protein which arises from protein misfolding, and there are actually several examples in everyday typical cells where protein misfolding occurs and you have insoluble proteins, and that can actually change the osmolarity of a cell and also cause a cascade of negative effects. Now, APP, or amyloid precursor protein, is exactly that. It's the precursor protein to an amyloid form, to an amyloid. Now, here we actually have a simple diagram of the phospholipid bilayer. Now, APP is an integral membrane protein, so it exists in the phospholipid bilayer inside a cell, inside the brain. And what happens in the Alzheimer's disease is when you have an incorrect cleavage of APP. Now, this is a simple example, a simple diagram, of APP, and you have the different cutting sites. Now, usually, alpha cleavage is, the peptide bonds are hydrolyzed by alpha secretase, an enzyme. And alpha secretase gives rise to APPS alpha, and then further cutting happens by gamma secretase. So gamma secretase, the enzyme, picks up where alpha secretase leaves off, and you have no negative effects which occurs. And what is characteristic of Alzheimer's disease is actually when you have beta cleavage by, conveniently, beta secretase. And beta secretase actually cuts the APP protein along an abnormal path, or where the on the polypeptide, and actually gives rise to the Alzheimer's disease. And gamma secretase now does not know where to cut because it usually recognizes when alpha secretase cuts. Now it doesn't know where to cut, so it actually cuts it twice, the APP protein, and it gives rise to two insoluble proteins called A beta 40 and A beta 42. And A beta 42 is what's characteristic in senile plaque formation or Alzheimer's disease. Okay, so as I mentioned before, A beta 42, which is the insoluble protein formed by incorrect proteolytic cleavage by beta secretase, results in actually beta pleated sheets, which you would know from lectures, the structure. And those result in the beta amyloid plaques, which are the insoluble proteins which aggregate in the brain, which is the characteristic formation of Alzheimer's disease. Now, what we're going to talk about now is actually neurofibrillary tangles and the protein tau, or NFT. Now, the tau protein is actually important because it's what holds together the microtubules in a cell. Now, the microtubule is what is important in transporting a cell to carry proteins and whatnot from one part of the cell to the other. Now, you actually have, as a result of the amyloid plaques, you have modification of tau proteins by hypophosphorylation. hyper meaning over or abundantly, and phosphorylation you would remember from biochemistry. Now, hyperphosphorylation of the tau protein results in NFTs, which is what we're going to discuss now. Now, when this is a simple diagram of a microtubule, and you can see here, exaggerated in red, is the tau protein, which holds together the microtubule. Now, when you have hyperphosphorylation of the tau protein, it isn't able to function anymore in what it was designed to do, which is hold the microtubule together. So, as a result, you have the microtubule actually being degraded, and then you have these tau proteins forming aggregations. So, follow along, the microtubule is first held together by tau, and because of hyperphosphorylation, the microtubule falls apart. And because of that, you have these, transport in the cell breaks down, in the cell brain particularly, and you have the tau aggregation, and then you have the formation of neurofibrillary tangles, which are insoluble. Now you actually have two negative effects, which is the protein transport being transport in the cell being affected, and then you have the formation of neurofibrillary tangles. Now, the problems with the plaque formations resulting from amyloid plaques and from NFTs is that you have inflammation of the neuron. So the brain would imagine these amyloid plaques, these insoluble nodules in the brain, as something negative, and it will inflame the area, which you remember is how the body deals with infections and whatnot. So you would have inflammations of the neuron, and you would have hyperphosphorylation of tau, which would result in transport in the cell being degraded and the formation of NFTs. And these two key protein misfolding problems is what gives rise to Alzheimer's disease in the brain. And this happens over a slow process, as I mentioned in the first part of the tutorial, and as it moves from the hippocampus of the brain from one area to another, affecting different centers in the brain, with short-term memory, as a recap, short-term memory and logic and solving problems, and then expression of emotion and control, and finally the oldest memories are affected. And in the last stages of Alzheimer's disease, we would have a literal loss of a person's ability to control simple muscular coordination and the involuntary muscle contractions, and that is how you result in death. It is a very slow process because, as you would imagine, it is these little tiny aggregations which must occur and form little nodules, and then the brain will try to attack and inflame the area. So it is actually quite a long process, and it is a very serious process because you have it's not only about proteins, but you actually have a physical, psychological effect on a person, which is what is very traumatic for most families who have to deal with people with Alzheimer's disease. So I hope this helps. Once again, the protein misfolding reasons why you would have Alzheimer's disease being developed. So thanks for your time.
Come in, Miss Pascal. Who are you? Miss Pascal, my receptionist. Anyway, I'll have to have this. I have to go and pick up my wife and my son and the pediatrician.
Dad, your wife?
Dad, my wife, Stephanie. Yeah.
Dad, long time